Today, I would like to take you on a journey through a forest, the one that has been transformed by bar beetles, yet holds remarkable survivors. My work focuses on Norway spruce and its fate during severe Ipsipographus outbreaks. This is a story of what it means to survive biologically, chemically, and genetically when everything around falls. If you fly over or walk across the Šumava National Park at the Czech-Austrian-German border, you will see kilometers of dead spruce. It's a ghost forest. The outbreak left behind kilometers of dead wood. It was a really devastating event. It began in the 1990s, and in some places it is still ongoing. Here is a helicopter flight picture from 2015. 12,000 hectares of forest were destroyed, or spruce forest were destroyed in the Bohemian forest area, the largest contiguous forested area in Central Europe. However, across the devastation, here and there, one green tree still stands. Less than 1 in 100. We call these trees the last trees standing, or simply LTS. Why did these trees leave when all neighbors died? That was the question that began this research, and one that still fascinates me. So many people might say that survival is just luck, that LTS were random remnants, but I felt that there was a pattern to be discovered, and our first clue came from spatial analysis. We mapped all surviving trees and ran the optimized hotspot analysis, which indicated that survivors are significantly clustered across the study area. So survival was not random. There was already something systematic protecting these trees. Then nonlinear regression models showed using the nonlinear logistic models, we started to see the excuse me. Smaller size increased survival. Trees that grew in less dense stands had a higher chance to survive. Also, more shade and long-term moisture availability all increased survival, and these positive effects were enhanced even more in water abundant conditions. So our results in Bohemian forest indicated external conditions for survival. However, we were interested in what's happening inside of the tree itself. And to explore that, we turned to France, where in 2003, there began another Ipsipographus outbreak. It was in Vosges Mountains in La Chaux-de-Fonds Nature Reserve. We conducted a mechanical wounding experiment to investigate phenolics as a part of tree chemical defenses. Out of 5 identified phenolics, catechin was the only one that exhibited significant differences between surviving and dead trees, and it was higher in dead trees. Now, the relationship between catechin and survival was nonlinear. Survival probability increased rapidly and then leveled off. However, we don't really know what's happening on that part of the curve, on the decreasing part. And for one, it could be because we didn't have enough high catechin survivors, or else it could be because catechin is a small phenolic that can be used as a building block to biosynthesize larger, more complex phenolics, such as condensed tannins. So higher catechin not necessarily means weaker defense. It could mean instead that catechin is being used to produce other defensive compounds. The logistic models also indicated that the farther away we get from the previously colonized trees, the higher chance to survive, which is expected. Ground defoliation and sun illumination of stem had negative effects on survival. We also found that the crown told a clear story. Both total and primary ground defoliation were highest in the trees that were the primary targets of the beetles. Another very important indicator of tree health and stress was the juvenile crown ratio. That's the very top part of the stem responsible for tree vertical expansion. And it was the lowest in primarily attacked trees. Back in the Bohemian forest, we wanted to see how survivors lived before the outbreak. By analyzing tree rings, we reconstructed growth histories, and the results were striking. Decades before the outbreak, 20, 40, 60, and even 80 years prior to disturbance, LTS were growing more slowly than reference trees. It seems that survivors invested more in maintenance and less in current growth trading speed over endurance. You could compare these trees to conservative investors who prioritize savings over current spending, and when the crisis came, this cautious strategy paid off. So more investment in protection and less in current growth suggested that there must have been some internal pattern or some driver encouraging these outcomes from within. And to explore that, we sampled phloem of these trees to analyze the chemistry of terpenes and non-structural carbohydrates, and also genome for DNA analysis. Using a 50K SNP chip and discriminant analysis of principal components, we identified weak yet significant genetic structure among surviving and reference populations. Using genome-wide association studies, we found that there were 12 SNPs, here above the line, 12 SNPs, significantly associated with survival. And out of these 12 significant SNPs, there were 3 that had biological functions in Arabidopsis thaliana orthologs. And all these 3 functions were related to tree stress responses, being it DNA methylation reactions or growth interruption in response to stress. So definitely, we clearly see now that there is a genetic fingerprint behind resistance. Chemically, the story continued. We inoculated methyl jasmonate to study terpenes as a part of tree-induced defenses. And 3 weeks after inoculation, we collected 3 samples for treatment and 3 samples half a meter above for control. Targeted gas chromatography mass spectrometry identified 56 terpene compounds that are here classified in mono-sesqui and diterpenes. And despite being statistically insignificant, we see a biological pattern that monoterpenes are higher in LTS, it's green color, while sesqui and diterpenes are higher in reference trees. The total chemical profiles were not dramatically different. However, a few compounds stood out, namely alpha-terpinolene, gamma-terpinene, and neobiotic acid were consistently higher in LTS. And these concentrations are in uninoculated phloem. That means that survivors have had upregulated levels of constitutive defense. That is an omnipresent defense that is always there in tree. It doesn't need mechanical wounding to be upregulated. And our possible explanation that maybe many years when the outbreak occurred, the surviving trees upregulated the level of constitutive defense. And for some reason, maybe due to epigenetic changes, they managed to maintain this elevated level up until now. But that's just a suggestion. We don't really know exactly. Another interesting pattern emerged when we analyzed non-structural carbohydrates. And it turned out that sucrose concentrations are higher in surviving trees, while starch is significantly higher in reference trees. It seems that LTS prefer to store energy in ready-to-use form, while others lock it away. Random forest models and nonlinear regressions showed us that faster long-term annual growth decreases survival probability, while sucrose concentration increases it. And the effect of neobiotic acid on survival probability was nonlinear, and it decreases and then it levels off. And this recurring pattern suggests that chemical defense can be effective up to an optimal threshold, beyond which extra investment of energy yields little or no additional protection. Now to the newest part of our results, those that haven't been presented before. Of course, tree chemistry is only half of the dialogue, and the other half are bark beetles. So the next question was, do all these compounds have real effects on bark beetles? And to explore that, we conducted electroantenography, which indicated that 3 volatile compounds, alpha-terpinolene, gamma-terpinene, and sabinine hydrate, elicited active responses on bark beetle antenna. Very good, but no surprise, already known from the previously published literature. The other 2 compounds, methyl dihydroabiatal and neobiotic acid, did not elicit any responses on the antenna, but that's not because they were unimportant. That is because they were large weight, non-volatile molecules, and a different testing method was needed to investigate their effects. So we had to conduct a feeding bioassay we brought in the beetles into the lab and we asked the beetles themselves what they thought about neobiatic acid. However, to design a feeding bioassay from scratch was no joke, it was a real challenge. And we spent weeks and weeks of trial and error in the lab, trying to figure out how to prepare the diet that was neutral yet attractive to the beetles to initiate tuning and feeding. For how long the beetles should be starved behind the test, at what light and temperature conditions they behave most naturally, how to get the diet into the tubes, and crucially, how to get those micro concentrations of neobiatic acid into the diet, avoiding any bias from the solvent. So as you can see, each and every detail was important, and it was a decision-making process as a result of trial and error. The team did a really great job coming up with a working bioassay feeding protocol in just 2.5 months. And I should say it's a really great result compared to our previous colleagues' experience when it takes at least 4 months to make a feeding bioassay working for each sporophis. So great job, many thanks to the team. Once the whole setup was ready, we used 30 males and 30 females for each of 6 treatments, ranging from no compound to double the peak concentration observed in surviving trees in the field. But before I show you the results, okay, so we monitored how far each beetle was tunneling into the tube and how much they fed on the artificial diet. Before I show you the results, for a little bit of a break for you, I would like to show a short clip to give a feeling of what our lab and field work looked like. Just for your information, the majority of beetles survived as the result of this experiment, and we let them all go after that. But that's a minor thing if someone is interested. Yeah, the results were compelling. Both males and females under double concentrations of neobiatic acid tunneled to the least and significantly less than in both controls. The feeding behavior was more variable. However, we see a very similar trend that under highest concentrations, high and double concentrations of neobiatic acid, both males and females, feeding behavior was deterred. Females responsible for laying eggs and building galleries were slightly less affected by neobiatic acid, which makes a biological sense. So the chemical signal that we identified in the trees translates directly into the beetles. And tree chemistry is not just passive, it talks back to the beetle, and that's the key finding. It's very important now. When we step back, a coherent picture begins to form. Survival is not random. It's the result of interactions between external and internal factors, all interconnected. More moisture and shade help. Smaller size and slower growth help. Specific compounds such as diterpene, neobiatic acids, and phenolic catechin all increase survival. And behind it all, there is a subtle yet real genetic foundation. To me, this is a hopeful story. Even amidst the most extreme disturbance, nature keeps a few seeds of resilience alive. The last three standing are not just accidents. They are archives of survival carrying clues to how forests might adapt in a warming world. Tree survival is not random. It's the story written in growth rings, in molecules, and in the genes. It's the forest's way of remembering and adapting to a crisis. This research has been growing with me over 7 years, and I would like to say a big thank you to all the people and teams I've been lucky to work along the way. It was a deeply collaborative journey across borders, disciplines, and landscapes, where sometimes moving 100 meters an hour was an achievement. It was a hard, slow, and complex research. And maybe it's not the fastest path for a career, but it is an honest one, true to myself and to my conscience. Our field team are real heroes, moving in areas where every step could reveal an abyss or a bear. In the Poljana area of Slovakia, there are 60 bears over 10,000 hectares, yet they keep going. And the bioassay too was a true team effort, and I would like to give a special thank you to Janka, who is here today, for her dedication in the lab, and to everyone who went out twice a week to collect fresh beetles, and to those who helped a lot with the technical work in the lab. We continue searching for LTS trees now across Slovakia and beyond within the Prosomics project, trying to read what they have to tell us. There's still much to be explored, and every tree that stands alive among the dead reminds us that resilience is possible. Thank you.